Not gonna lie, no matter how you draw it out, the pentose phosphate pathway, especially the non-oxidative phase, is basically always going to look confusing. And my goal isn't for you to understand exactly what's going on at every step and memorize what enzyme does what, but my goal is to kind of walk you through the logic of it, help you go through this pathway at least once to follow the carbons and see what's happening. And then you will have a better appreciation of this pathway and how it is interconnected with a lot of other metabolic pathways. So the pentose phosphate pathway is really important. As we've discussed, it's going to give us our NADPH, which we need for things like making lipids and steroids and regenerating our glutathione, our antioxidant, so that we can combat reactive oxygen species. And it's also going to give us our ribose that we need for making nucleotides, which we need for DNA and RNA and some cofactors and things like that. So today we are going to walk through this complicated looking diagram, going step by step. Hopefully you can be following along with some Legos. So in class we did this. We started with 6 glucoses and then we took them through and we moved around paper clips to show the position of the carbonyl. And we used yellow tape to show phosphates. And so that was a really helpful way to kind of visualize this kind of carbon scrambling phase. So just to review really quickly before we dive in, basically what happens in the pentose phosphate pathway is we have 2 parts. We have an oxidative phase and we have a non-oxidative phase. These are going to basically interconnect with our glucose metabolism, with our glycolysis and our gluconeogenesis at this like fructose 6-phosphate and glyceraldehyde 3-phosphate steps. We'll see that in the oxidative phase of the pentose phosphate pathway, we have this basically committed regulatory step where we have our glucose 6-phosphate dehydrogenase. That's basically going to oxidize our glucose 6-phosphate to give us 6-phosphogluconolactone. That's going to generate an NADPH, but we don't lose any carbons. In gluconolactinase is basically just going to hydrolyze that. Then we're going to have 6-phosphogluconate dehydrogenase where we generate another NADPH, and here we're actually losing carbons. This part was going to be irreversible. We generate 2 NADPH and we lose a carbon. And now at this phase, we have multiple fates. We can take things through the non-oxidative phase where we basically take that ribose 5-phosphate. We can convert it to ribose 5-phosphate if we want nucleotides, or if we want to kind of repeat the whole cycle because we still have those 5 carbons left. Now what we can do is we can take that ribose 5-phosphate, split it into both ribose 5-phosphate or convert it to both ribose 5-phosphate and xylulose 5-phosphate, scramble those up with a bunch of reactions and enter them back into the glycolysis pathway, either to take them down for energy, or if we want to keep this pathway going to get more NADPH, we're going to interconvert them back into glucose. So we're going to follow along and see how this is actually going to happen, as well as look at some kind of points at which we can choose which way we want to go. Again, hopefully you can be following along with your Legos to use a different color Lego for each of the initial starting glucoses. And then you'll see that we're basically going to account for all of the carbons if we start with 6 glucoses. So by starting with 6 glucoses, we start with 6 5-carbon sugars, and we're going to end up with 5 6-carbon sugars. We just need to start with 6 so that the stoichiometry works out. But it's not like your cells always have to start with 6, especially because we can imagine that they're taking in, that they can be interconnecting with taking in fructose 6-phosphate and glyceraldehyde 3-phosphate that we generate through things like glycolysis, through things like lipid breakdown. So all sorts of different ways in which we can get these intermediates, we can funnel into this non-oxidative phase of the pathway, which remember is how we generate our nucleotides if we want those, or how we can kind of basically scramble things around in order to make more glucose 6-phosphate, which we can then oxidize in order to give us our NADPH. Whew, that was a lot, and we haven't even started the tracking. So let's go through the tracking because I do think it helps to see what's going on once. So by kind of color coding things, you can see what's going to go where. The way that I have this drawn out is that each of these filled circles, except for these ones with an O, these are going to represent carbons. And the bold position is going to represent the position of a carbonyl. So if we have one on the end, well, what do we have there? We have an aldose because we have an aldehyde sugar. And if it is in the middle, and without sugars, these are going to be in like the second from the top position, so the C2. If our carbonyl is there, well, now we have a ketone sugar, we have a ketose. We can further classify the sugars based on how many carbons that they have. So we have glucose, which is our 6-carbon aldose. We can phosphorylate that, and that's still our glucose, but now we have glucose 6-phosphate. If we basically then move the position of the carbonyl to that C2, well, now we have a ketose, we have our fructose. If we want, we can phosphorylate that again to give us fructose 1,6-bisphosphate. This is what we would do if we wanted to go to glycolysis. We would basically take that glucose 6-phosphate, isomerize it to our fructose 6-phosphate, and then rephosphorylate it again, and then go through aldoses. The reason why I included it here is because we'll actually see that in order for the carbon scrambling for the stoichiometry to work out, we're actually going to run the aldolase reaction in reverse. Remember, that's a reversible reaction where we can now basically take glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate, dihydroxyacetone phosphate, which we'll get from our, we can see how we can get that from the pentose phosphate pathway. We can interconvert those back in order to give us this fructose 1,6-bisphosphate, which we can then basically dephosphorylate to give us our fructose 6-phosphate, isomerize to give us our glucose 6-phosphate, and then take that through the pentose phosphate pathway. But if we just start with thinking about our glucose and going through the oxidative phase, rather than thinking about this part, which was kind of to regenerate the glucose, if we just think about, okay, we start with the glucose, we trap it with our hexokinase, we've got this glucose 6-phosphate. If we then take our glycerol 6-phosphate dehydrogenase, our G6PD, and oxidize it, this is going to produce NADPH and give us 6-phosphogluconolactone. Note that we could have drawn the glucose in the ring format here as well, but for simplicity's sake, I'm drawing these in the linear format, except for the 6-phosphogluconolactone, because, well, that one has to be a circle, based on like the definition of a lactone, and you can't really draw it otherwise. So this is our 6-phosphogluconolactone. You can see that we've had an oxidation reaction. And because we were basically reducing NAD plus, P plus to give us our NADPH. We can now hydrolyze that with lactonase. We get 6-phosphogluconate. And so here we have a carboxylic acid at the end. And this is our 6-phosphogluconate, which now we can dehydrogenate. We can use 6-phosphogluconate dehydrogenase. Now we're going to basically do another oxidation reaction, but here we're actually going to be, not only do we generate NADPH, but we're also losing a carbon. So from here, all of these sugars have been hexoses. They've been 6-carbon sugars. Some of them were aldoses, some of them were ketoses. This one was like a carboxylic acid. But here what we have is now we have 5-carbon sugars. So we have pentoses. What we get originally is this ribose 5-phosphate. So the end of the oxidative phase gives us our ribose 5-phosphate. Everything up to here was basically going to be, this was like the irreversible phase. And now we're in this reversible phase where we have multiple different fates and we can kind of just scramble the carbons around based on what we need. This ribose 5-phosphate, we can see that we have a ketose. So it has the ketone so it has a carbonyl at the C2. What we can do is we can either kind of move it and make an aldehyde, an aldose. That would we could use ribose 5-phosphate isomerase. So remember, an isomerase is an enzyme that kind of just moves around the position of things. And it's going to basically move around the position of that carbonyl from the C2 to the C1. Now we have an aldose, we have a 5-carbon sugar, so we have a pentose. Okay, and we have a pentose that's really important, this ribose 5-phosphate, which is what we can use as a precursor for making our DNA and our RNA and some cofactors. We can also, however, so basically if we wanted to just make nucleotides, if we really wanted to make nucleotides, we would take all of this ribose 5-phosphate that we generated from this step, and we would basically go funnel it off into in order to make nucleotides. But then we want to be able to run this cycle again in order to generate more NADPH. So if instead of wanting nucleotides, we care more about our NADPH, well, now what we want to do is we want to basically take this 5-carbon sugar that we have, this pentose, and we want to take, so if we did this 6 times, we would end up with 6 5-carbon sugars. We now want to scramble those around in order to give us 5 6-carbon sugars. So basically what we're trying to do is we want to take these 6 5-carbon sugars, scramble them up to give us 5 6-carbon sugars that we can then do this process again. Again, to make the stoichiometry work out, we'll have to feed more into the pathway. But we can basically think about, if we think about in terms of 1 glucose, per glucose, we're getting 2 NADPH and 1 5-carbon sugar. If we still have 5 carbons left over, where now if we were to convert that to a 6-carbon, we could basically do this again. So if you think in the terms of like how many per carbon, you're generating 2 NADPH. And so if you think about a glucose molecule, here you should be able to generate 12 NADPH from a single glucose molecule if you were to keep doing this again and again, provided that you could feed intermediates into the pathway to keep regenerating the glucose 6-phosphate. And that's how we kind of get the carbon scrambling, just to account for kind of piecing together the skeletons of different molecules from either the oxidative phase or from glycolysis in order to piece together glucoses from our leftovers in order to run the cycle again, which we can do, especially if we really want that NADPH for our ROS fighting, our lipid and steroid production, etc. So when we do this, we're going to see a bunch of different kind of like pieces of the skeletons as we rearrange things. To start things off, we can't just start with this ribose 5-phosphate. We also have to make this ribulose 5-phosphate, the xylulose 5-phosphate, which we make using our ribulose 5-phosphate epimerase. So in epimerase, well, here you're actually changing like the configuration of something. You're kind of changing whether one of these OHs is sticking to the right or to the left. So if we look at the C3 position and compare it to our ribulose 5-phosphate, if we just look at the simplified outline, we can see that both of these are going to be ketoses. So it looks like nothing's changed, but if we look at the actual carbon, like the structures, we can see that at the C3 position, we go from being on the right to being on the left with our OH. So these are different. These are epimers because they basically vary at 1 chiral carbon center. So these are our 5-carbon sugars, our pentoses. We'll also see that basically we can mix and match, take 3 carbons from one thing and add it to something else, or take 2 carbons from something or add it to something else. This is going to leave us with some 7 carbons and some 4-carbon sugars. So our 7-carbon sugar, what we'll see is cetoheptulose 7-phosphate. And so this is going to be our 7-carbon sugar. And then we have this erythrose 4-phosphate, which is going to be our 4-carbon sugar. And then so that would be our tetrose. And then we had these 2 trioses. We saw these 3-carbon sugars, our glyceraldehyde 3-phosphate. So here we have an aldehyde, and our dihydroxyacetone phosphate. Well, here we have a ketone. So we have a ketose and we have an aldose. And we can interconvert those using triose phosphate isomerase, which is that enzyme that we saw in glycolysis. We also saw aldolase in glycolysis. And we'll see that here, basically, we're running things kind of the opposite direction that we would be doing, and we can regenerate glucose. Note, however, that this is not going to be gluconeogenesis. So gluconeogenesis, the neo means new. And basically what's going to happen is that you're making glucose new from non-carb sources. So from like pyruvate, from non-sugars. Here, we're basically we're mixing and matching sugars to make glucose. So we wouldn't call this pathway gluconeogenesis. And we're not ending with glucose, we're ending with glucose 6-phosphate, which we can then take back through this pathway. So remember that in order to make glucose, you actually have to have glucose 6-phosphatase in order to like free up the glucose to release it. And only your liver and kidneys are going to be able to do that. So your tissue, like basically all your tissues would be able to do this pentose phosphate pathway. They can make this glucose 6-phosphate that they can then feed through the pathway and make NADPH depending on their needs. So let's now look at that carbon scrambling and kind of see what's trying to track the carbons and see what's going where. So again, we start with our 6 glucoses. So we just start with 6 so that the stoichiometry works out. If we think about them each as being a different color, it's going to help us track the fates of each of these carbons. In order to trap these, the sugars inside the cell, we have to phosphorylate them. This is going to be done with our hexokinase, which remember in our liver, we call that our glucokinase. And in our liver, we have that version that's going to have a lower affinity so that it doesn't hog all the glucose and so that it can let out glucose that it makes. And it's also going to not be inhibited by glucose-6-phosphate so that it is able to take in a lot of sugar because it can store it as glycogen. And it's kind of like if the other tissues already, because the other tissues have a higher affinity hexokinase, and if they already kind of like took what they need, we want the liver to be able to take in the rest so our blood sugar doesn't stay too high. So that's our cool stuff about our liver glucokinase. But now let's just talk about no matter which hexokinase isozyme we used, we now have this glucose-6-phosphate. By adding this phosphate, we've kind of like trapped it in the cell. And we've also trapped all of these other smaller intermediates so that they're all going to be phosphorylated. Okay, so we have this glucose-6-phosphate. Now what we're going to do is we have those multiple fates that we have to consider. We could take that glucose-6-phosphate, and if we wanted to do glycolysis, we could use phosphoglucose isomerase to make fructose-6-phosphate. This would be great if we want energy, because remember glycolysis, we're going to break things down to pyruvate. We can take that pyruvate through the citric acid cycle, generate lots of NADH and FADH2. We could then take those through the electron transport chain and oxidative phosphorylation and make ATP. Awesome. Okay, but what if we care less about that and we care more about storing the glucose? Well, now we could use phosphoglucomutase, make glucose-1-phosphate, store that as glycogen. Remember, that's going to be happening predominantly in our liver and in our muscles. But we don't want to worry about those things right now. We're going to worry about this pentose phosphate pathway. And the kind of key regulatory step here, the step that's going to commit us to this pentose phosphate pathway, is going to be this glucose-6-phosphate dehydrogenase step. In this step, we're going to generate NADPH from our NADP+. So we're reducing NADPH. We're oxidizing our glucose-6-phosphate. This is the first step of our oxidative phase. So if we go back and look at what's happening here, we're generating this NADPH. And again, because we start with 6 glucoses, we're going to get 6 NADPH. And we're also going to be left with 6 phosphogluconolactones. Okay, so if we go back and we look at what these actually are, we've basically taken this glucose-6-phosphate. Now we have the 6 phosphogluconolactone. The next step we're going to do is we're going to kind of break that open. And now we're going to get our 6-phosphogluconate. So see that we still have a 6-carbon sugar, but now we have a carboxylic acid at the end, as opposed to having just an aldehyde at the end. So that we can see that we've basically undergone this oxidation step. Okay, so that was that. Now let's see what happens next. We have this 6-phosphogluconate, and we're going to basically do another oxidation to give us 6 and more NADPH. And we're actually going to decarboxylate. So we're going to end up with 6 CO2. So we're losing a carbon from each of these groups. And we're ending up with, now we're going to have 6 5-carbon sugars. More specifically, we have ribose 5-phosphate. So we have 6 5-carbon sugars and 6 CO2s. We can see that when we go through glycolysis, or when we go through like the citric acid cycle, when we lose a C, we're basically losing the opportunity to get energy. In this pentose phosphate pathway, in no steps of the pentose phosphate pathway are we directly generating ATP. And in fact, we're losing the opportunity to generate ATP because, well, if we scramble these carbons back up, we get 5 glucose-6-phosphates. We could take those 5 glucose-6-phosphates through all of our metabolic pathways and get ATP, but we would only get the ATP amount worth 5-glucose-6-phosphates rather than 6-glucose-6-phosphates. So we're not going to get as much energy if we were to go and use these for energy. So why would we do this? Well, we do this because we want the NADPH. And this NADPH is going to be super duper important. How does the cell know kind of like what to do when? Well, if we look back at this key regulatory step, this glycerol 6-phosphate dehydrogenase is going to be regulated by the amounts of our NADPH. It's going to be inhibited by high levels of it and stimulated by low levels of high levels of NADP+, so kind of like the opposite. And we're also going to have steps that are regulated directly by our reactive oxygen species. So go back and review the Pentose Phosphate Pathway regulation part of the video if you're interested in that. But for now, let's think about, we end up with this ribose 5-phosphate. So we have these pentoses, and we basically have this carbon at the second position, the aldehyde at the second position, so we have ketoses. And now what we're going to do is we're basically, if we wanted just nucleotides, we would take them all, we would use this ribose 5-phosphate isomerase, and basically just take these ketoses and make them aldoses. So move the position of the aldehyde from the C2 to the C1. This would be done with our ribose 5-phosphate isomerase, our RPI, and then we could take that ribose 5-phosphate and you can use it for nucleotides. If we really wanted nucleotides, so we wanted NADPH and we wanted nucleotides, what we would do is we would just do this. If we wanted them like equal amounts, we would do this. We would take all of this ribose 5-phosphate and convert it all to 5-ribose 5-phosphate and use those for nucleotides. But now we wouldn't be able to get any more NADPH. So all that we would have generated would be 12 NADPH, so 2 per glucose that we started with, as well as per glucose that we start with, we would get 1 ribose 5-phosphate. If, however, we care more about the NADPH, well, now we want to take these 6-5-carbon sugars and basically scramble up the carbons to give us more glucose 6-phosphate that we can then enter through the pathway again. And this is where we get into that confusing carbon scrambling phase. Note that in this carbon scrambling phase, basically all of these reactions are going to be reversible. So we can interconvert these carbons, go back and forth, go in and out of glycolysis. So one of the nice things about the pentose phosphate pathway is it kind of lets you see how these pathways interconnect. But one of the not so nice things about the pentose phosphate pathway is that it's going to look really, really confusing. So try to track the positions of the carbonyls, as well as use the coloring scheme to help you follow the carbons. And hopefully, again, you can be following along with your Legos, because I think that's a really helpful way to see what's going on and kind of visualize it more. But let's start and just kind of see how we can take these 6-5-carbon sugars and transform them into 5-6-carbon sugars. What we're going to do is we're going to use ribose 5-phosphate isomerase, so that when we saw that we can make ribose from our ribose 5-phosphate, we're going to make 2 riboses, and then we're going to take the other 4 that we have left over, and we're going to use this ribose 5-phosphate epimerase. And basically, we're still going to have a ketose, but we're going to swap the position of the third carbon, the OH on the third carbon. Basically, we're just going to change at this chiral center. We're changing a single chiral center, so these would be epimers. Once we've done that, well, now we have these ketoses and aldoses. Both of them are 5-carbon sugars. If we take 2 5-carbon sugars, that's 10 carbons total. We can swap things around in order to get a 7-carbon sugar and a 3-carbon sugar. How we're going to do this is using transketolase. So transketolase is going to transfer 2 carbons from a ketose to an aldose. In this case, our ketose is going to be our xylulose 5-phosphate, and our aldose is going to be ribose 5-phosphate. So what we're going to do is we're going to take one of these X5Ps and one of these R5Ps. We're going to move 2 carbons from our X5P onto our R5P, and this is going to leave us with said-uheptylose 7-phosphate and glyceraldehyde 3-phosphate. So if you imagine taking this orange one and this purple one, moving 2 carbons from the orange to the purple, Well, now what we end up with is we're going to get a longer ketose and a shorter aldose. So our ketose became a shorter aldose, and our aldose became a longer ketose. We do this twice. So here we're doing, you can see that we're doing it with like this green and this brown. Again, this is the exact same reaction, and we're generating a 7-carbon sugar and a 3-carbon sugar. We started with 2 5-carbon sugars, and so we have all of our carbons accounted for. We just swap things around. Now we're going to take these products and we're going to swap things around even more. We're going to use this enzyme called transaldolase. Now, transaldolase is going to do something similar, but it's going to transfer 3 carbons from a ketose to an aldose. So both of these are going to be moving carbons from a ketose to an aldose, making a longer aldose and a shorter, or longer ketose and a shorter aldose. You can remember aldolase is going to transfer 3 carbons because if we think back to the aldose step in glycolysis, well, here what we're actually doing is we're taking a 6-carbon sugar and breaking it into 3. And if we think about the reverse, we're taking 2 3-carbon sugars and adding them together. So if that's moving 3 carbons, and so here we're moving 3 carbons, but we're kind of like doing it trans. So we're basically adding from one sugar to another sugar rather than just combining 2 3-carbon sugars. So let's if we think about this orange one, and now we have this one that has purple and orange. This is going to be our ketose. This is our aldose. We can transfer 3 carbons from the ketose. So if we think about taking these top 3 carbons and transfer them onto this aldose. This is going to give us with a 6-carbon ketose, our fructose 6-phosphate, and we're also going to be basically left over with this 4-carbon sugar, this erythrose 4-phosphate. And this one's going to be an aldose. So note that glyceraldehyde 3-phosphate, our GAP, and we had here could have come from glycolysis, or it could go into glycolysis, depending on our needs, but then the stoichiometry wouldn't work out. This fructose 6-phosphate, we can basically transform it directly into glucose 6-phosphate using phosphoglucose, or also known as phosphohexose isomerase. That's going to basically give us our glucose 6-phosphate, which is what we're trying to get. So this one's already kind of like done, but here we're left with this 4-carbon sugar, this erythrose 4-phosphate. And again, we can imagine doing it the same reaction with the second set that we had. So we take these 3 carbons from our cytoheptulose 7-phosphate, plop them onto glyceraldehyde 3-phosphate. We get a fructose 6-phosphate. We can directly transform into glucose 6-phosphate, and we would get a erythrose 4-phosphate, so another 4-carbon sugar. We only did that reaction twice when we were up here. So we used 4 of our 5-carbon sugars. We still have 2 xylulose 5-phosphates left over. Because remember, we did 2 of the ribose 5-phosphate and 4 of the xylulose 5-phosphate. So we did 2 of the RPI, 4 of the RPE, and then we used 2 of each. So we're left over with 2 of the xylulose 5-phosphates and 2 of these erythrose 4-phosphates. So a 5-carbon sugar and a 4-carbon sugar. We have 2 of those. So we have 10 carbons plus 4 carbons. We have 18 carbons. We should be able to make 3 glucoses. How are we going to make 3 glucoses? Now things get a little more complicated. We're going to have to go through several different reactions. We're going to start with transketolase. We take that xylulose 5-phosphate that we have left over from our RPE step, and we take that erythrose 4-phosphate that we have from our transaldolase step. We're going to use transketolase again, but this time we're starting with different things. The same thing's going to happen, though, is that the transketolase is going to transfer 2 carbons from a ketose to an aldose. In this case, our ketose is going to be our xylulose 5-phosphate, and our aldose is going to be our erythrose 4-phosphate. Xylulose 5-phosphate, so we're going to transfer 2 carbons from our xylulose 5-phosphate onto erythrose 4-phosphate. This is going to give us our fructose 6-phosphate and a glyceraldehyde 3-phosphate. Remember that the fructose 6-phosphate could readily interconvert with phosphoglucose isomerase to give us our glucose 6-phosphate. So that would be taken care of. However, we're also left with this glyceraldehyde 3-phosphate, this gap. if we do this with each of these remember we had 2 xylulose 5-phosphates and we had 2 erythrose 4-phosphates. We combined those. We get 2 fructose 6-phosphates, which we convert to glucose 6-phosphate, and 2 glyceraldehyde 3-phosphates. Now we just have to take those 2 glyceraldehyde 3-phosphates and we have to combine them to make glucose 6-phosphate. We can account for the sugars. We have 2 3-carbon sugars, but we now also have 2 phosphates. If we were to basically put the gaps together, we would make a 6-carbon sugar. But rather than directly put them together, because remember, these are both going to be, at this point, we have 2 aldoses. When we join sugars together, we basically need an aldose and a ketose. And so what we're going to do is we're going to take one of these aldoses, this glyceraldehyde 3-phosphate, and we're going to use triose phosphate isomerase to make dihydroxyacetone phosphate, or DHAP. We do this using triose phosphate isomerase, which is the same thing that we are going to do in our glycolysis. So in glycolysis, we could use triose phosphate isomerase to interconvert between our glyceraldehyde 3-phosphate and our dihydroxyacetone phosphate. In glycolysis, if we wanted to go and break this down further, we would then use glyceraldehyde 3-phosphate dehydrogenase in order to generate 1,3-bisphosphoglycerate, which we can then take further. But that's not what we're going to do here. Instead, what we're going to do here is we're going to take that glyceraldehyde 3-phosphate, convert it to dihydroxyacetone phosphate, and then add a dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, kind of merge them up together using aldolase. So in this case, what we're doing is we're basically adding the dihydroxyacetone. We're adding this ketose to an aldose, and we end up with this fructose 1,6-bisphosphate, this F16BP, which again should be familiar to you from our glycolysis. And it's really close to fructose 6-phosphate. All we need to do is use fructose 1,6-bisphosphatase in order to kind of cut off that phosphate and give us fructose 6-phosphate, which we can then basically interconvert just like we did before to give us our glucose 6-phosphate. It's the same reaction that we were doing here, this phosphoglucose hexose isomerase, because this fructose 6-phosphate is just like the fructose 6-phosphates we got made over here. We just made it a different way. At the end of this, what we end up with is we've taken these 6 5-carbon sugars and we've made 5 6-carbon sugars. We've made 5 glucose 6-phosphates, and now we could go do this pathway again. We could take these glucose 6-phosphates, take them through the oxidative phase, scramble up a bunch of carbons, and keep this going. For the stoichiometry to work out, you have to start with 6 glucoses, because 6 times 6 is going to be 36. Then you're going to lose 6 carbons, so you get 30. And then if we then we left up with 5 5-carbon sugars, scramble those, we get back to 30. So we still have 30 carbons, which we could split up into 5 glucose 6-phosphates. But in order to kind of keep the stoichiometry going, you need another glucose. And so that's where it really gets confusing if you try to think about the stoichiometry. But I don't need you to worry too much about that stoichiometry, because really we can feed into and out of this pathway. And it's not like these molecules are just like going through this cycle and it's the exact same molecules that are going in and out and in and out at all the time. What we can have is we can basically have this fructose 6-phosphate be coming from glycolysis. We can have it be coming from glycogen that gets broken down. We can have it be coming all the way from, say, glycerol that then got interconverted and went through all this pathway to give us our fructose 6-phosphate. We can have this glyceraldehyde 3-phosphate becoming similarly from glycerol. We can have it becoming all the way down from pyruvate. We can have it be coming down from glucose. So we have all of these different ways where we can kind of take carbons in and out of this pentose phosphate pathway, depending on what we want. The key things are that the pentose phosphate pathway is going to allow us to kind of make 2 things of interest. It's going to allow us to make our NADPH, which we need for making lipids, which we need for making steroids, which we need for fighting oxidative stress. And it's going to make ribose 5-phosphate, which we can convert to ribose 5-phosphate. And that is going to be used to make DNA and RNA and coenzymes. So depending on how much we need NADPH and depending on how much we need ribose, we can alter how much we use kind of the oxidative phase or the non-oxidative phase. The oxidative phase is where we're generating our NADPH. So if we have a lot of oxidative stress, that's what we're wanting to do. We're going to want to use that as much as possible. In order to use the oxidative phase as much as possible, we recycle all of these carbon skeletons that we were left up with from the oxidative phase back through. So in order to, if we were to just run it fully oxidative, we just keep doing this over and over, we wouldn't be able to get any nucleotides. In order to make nucleotides, we have to actually use the ribose that we made. And if we use that ribose, then we can't put those carbons back through the cycle. But if we need nucleotides, then we can do that. If we need nucleotides, but say we don't need the NADPH, well, now it doesn't make sense to go through the oxidative phase, because in the oxidative phase, we're losing CO2. We're losing the opportunity to make energy. But if we were to just kind of like go from glucose, make fructose 6-phosphate, and glyceraldehyde 3-phosphate, we can then feed those into the pathway. We wouldn't be losing any carbons, so we would be able to make more nucleotides. We wouldn't be losing the potential energy. But if we want both the NADPH and the nucleotides, well, then we basically just take the glucose through the oxidative phase and through the non-oxidative phase. We only use the carbon scrambling in order to regenerate more glucose 6-phosphate, which we can take back through the pathway. But we're not going to basically be on net making any energy. We're not going to be making anything like that. Per glucose, we're only going to make two NADPH, so one here, and one ribose 5-phosphate. But if we want more NADPH, then we take the skeletons, we recycle them back through and take them through this oxidative phase. We can do that 6 times per glucose. Now, and then we said if we wanted just the nucleotides, instead of going through the oxidative phase, we basically take these intermediates from this glycolytic pathway and just feed them into the carbon scrambling phase to make our nucleotides, because all of this was going to be reversible. Now say that we said if we wanted both, we'd do the oxidative phase and then the non-oxidative phase, but take the ribose 5-phosphate out. And what if we want even more NADPH? We can feed things in from glycolysis at all the way, but at the top at this glucose 6-phosphate step. That would give us the maximum amount of NADPH that we could make. A lot of times, however, you do want both NADPH and you want the nucleotides. Why might that be? Well, let's think about when you might want them. Remember, we said that with the pentose phosphate pathway, with the oxidative phase, we generate our NADPH. And our NADPH is going to be really great if we want to combat reactive oxygen species, or if we want to make lipids and steroids. If we think about when we might want to make lipids, well, lipids, if we're going to regenerate, we're going to have rapidly dividing cells, maybe like cancer cells, or other cells in growing tissues. These are going to need to make lipids so that they can basically split their cells and have enough membrane to go around for everyone. So they're going to need that NADPH. But what are they also going to need? They're also going to need to replicate their DNA. For that, they're going to need nucleotides. And they're also going to need ribonucleotides because they need to make mRNA, which they need to make proteins. So they're going to have need for both. And so the pentose phosphate pathway in its entirety is often going to be used there. However, if we just have, say, reactive oxygen species, then we care more about the NADPH, and we just want to do all that carbon scrambling to keep the oxidative phase running. And if we just wanted nucleotides, we have some reason why we need to make a lot of DNA. Maybe it's pre-replication, or we want mRNA or something like that, or maybe we just have a bunch of NADPH already. In that case, we can then just take those intermediates from the glycolytic pathway and feed them through. If we want NADPH and we want energy, well, here what we could do is we could basically run the oxidative phase, use carbon scrambling in order to scramble together those skeletons, feed them back into the glycolytic pathway, take them down to make our ATP. So I know it looks like a lot, but hopefully you get the main concepts. All of this is basically just scrambling around the carbons the skeletons that were left over from the oxidative phase in order to generate more glucose 6-phosphate. You want to do that, you only need to do that if you kind of want to keep the oxidative phase going, or if you want these intermediates to feed back into glycolysis. But if you really just want energy from glucose, you're not going to use the pentose phosphate pathway. You're just going to go straight down. Hope that was helpful. Hope you could follow along with some Legos, and just don't get tied up with the details. Just focus on the key ideas.